So now we can go on with the second part of this interesting meeting. And on the agenda, we have Professor Tassula Proikas-Cezanne from the University of Tübingen, who will talk about the molecular function of WDR45 WIPI4 in autophagy. Please, Tassula, the stage is yours.
Thank you very much. I'm trying to share my screen now. And I hope it works if I don't hear.
Yes, ma'am, it works.
Thank
you are ready.
Thank you. So first of all, I'd like to thank the organizers for inviting me. It's an exciting conference and I learned a lot, really a lot yesterday and also today. So today I'd like to share with you our passionate interest in the WIPI genes, in particular the role in autophagy, health and disease. And here we have a current interest in BPAM. So before I start, I'd like to give you an introduction, very basic introduction to autophagy, starting off with what it means. It's a Greek word and it means self-eating. And autophagy is a catabolic pathway. So it really is a form of self-eating. And it secures eukaryotic cellular homeostasis. It defines the lifespan, because if we knock out autophagy genes, for example, in C. elegans, then lifespan is reduced by up to 1/3. But the problem may be with age that autophagic activity declines, and that gives rise to a variety of age-related human diseases that are sometimes correlated with metabolic reprogramming, like in tumor. Now in BPAM, we have a mutation of an autophagy gene, and that's WIPI4, and we'll come to this in a second. So autophagy is a catabolic pathway, meaning material from the cytoplasm is degraded through this process, but upon this degradation, autophagy-derived metabolites support anabolic functions. So here you see the cargo material that becomes sequestered in a membrane that is built de novo in close proximity to existing membrane systems, but is built de novo and expands and sequesters the cargo and closes up to build a double membraned autophagosome. And the autophagosome then fuses with lysosomes to acquire acidic hydrolases, because they're important to break down the cargo. And eventually the material can be used for recycling purposes. Now, basal autophagy occurs on a very low level constitutively. And that's very important, especially for non-dividing cells like neurons, where unwanted material is degraded through this process. And on top of basal autophagy, autophagy can be induced upon cellular stress, and nutrient starvation, glucose starvation is one of this inducing signal. And then we have two types of autophagy, how the cargo becomes sequestered in the autophagosome. It can either be selective, so depending on the cargo, or not, we'll come to this in a second, but the cargo is recognized that needs to be degraded, and the membrane forms around it. And then there's stochastic autophagy, meaning parts of the cell become sequestered, and thereby also the material that is there in this growing membrane, and then stochastically material is degraded. That basically keeps our cells young and, as I said, secures cellular homeostasis, or contributes to securing cellular homeostasis. So cargo can be really almost everything in the cell: proteins, lipids, membranes, ribosomes, nucleic acids, and importantly, entire organelles. So mitochondria, peroxisomes are degraded through this process. And that's the only process that takes care of the degradation of organelles in eukaryotic cells. And important for our session is also a process that is called ferritinophagy. And this is a process where ferritin is selectively degraded through the process of autophagy. And that became clear in 2014 when the so-called autophagy receptor, NCOA4, was identified in a proteomic approach. And this is depicted here. So this autophagy receptor communicates with a membrane, with a growing membrane. It's also called phagophore, whereby the autophagy protein LC3 binds this autophagy receptor, NCOA1, and this one specifically binds to ferritin. And thereby, ferritin is selectively sequestered in the autophagosome and is thereby degraded. So because this is a specialized form or a specific selective form of autophagy, it's called ferritinophagy. So how does it all start? Initiation of autophagy depends on the production of a phospholipid, and that's PI3P. And this is under the control of two conserved pathways, the AMPK pathway and TORC1 pathway. So the initiation signal to build the membrane, to sequester the cargo, is really the production of PI3P. And we became interested in autophagy only because we discovered years ago now genes and proteins that specifically bind this newly produced PI3P at the onset of autophagy. And we refer to them as WIPI, WD repeat protein interacting with phosphoinositides. It's a conserved family, it's an ancient family present in all eukaryotes. In human, we have 4 members, 1, 2, 3, 4, and 4. The gene is, as you know, referred to WDR45. Now, when we cloned the genes, we did homology modeling, and we found something interesting. We found invariant residues, so meaning residues that are identical from yeast to human. They're depicted in pink here. And then there were homologous residues that were also conserved. And when you flip this beta propeller, 7-bladed beta propeller, which became apparent through this homology modeling, to the side, you see that the invariant residues, they cluster and form a motif. And we then showed, and others, that this motif is specific for PI3P binding. On the other side, we have residues that confer protein-protein interaction. When we started off, we looked at WIPI1, endogenous WIPI1 with a raised antibody, and we observed that it forms puncta upon starvation. And those puncta are co-localized with LC3. And there we felt this family should be involved in autophagy. So it was the first beta propeller the first beta propellers that bind PI3P and that were connected to autophagy. We then went on and used this specific localization to autophagosomal membranes and established an automated high throughput analysis where we used initially WIPI1 to score for the autophagic status, and we can use a variety of cells, cell lines, and also primary cells. And we go about this that we conduct automated image analysis, acquisition, and then followed up by automated image analysis, where you can see a heat map here. And when we look at the individual fields, we can detect with the system nicely the cell, we can nicely detect the nucleus, and we can detect autophagosomal membranes. So we use this system for a variety of approaches to work on autophagy, on the process, to address the function of the WIPI proteins in autophagy, but also to monitor autophagy, meaning to measure the number of cells that undergo autophagy by measuring the number of punctures here. And other imaging systems confirmed that these indeed were autophagosomal membranes. So colocalization studies, of course, we did, but also types of electron microscopy. And what you see here is a freeze fracture immuno-EM where we used WIPI1 antibody and detected it on both membranes of the autophagosome. Now we have 4 WIPI proteins, 4 genes, and when you look at them, they look extremely similar. You see here WIPI1, 2, 3, and 4, and depicted in red are the crucial amino acids that are responsible or required, essential for PI3P binding. And initially, we addressed the question whether or not they have a redundant functions or not in autophagy. And what I will be showing you here is latest results on this approach. So again, you see here the 7-bladed beta propeller formed by all 4 WIPIs. You see they all bind PI3P. This is protein phospholipid overlay, and they all bind to autophagosomal membranes via PI3P. And that is shown here. So you see here 3D reconstruction of WIPI decorated membranes. You nicely see phagophore structures here. And you also see this nicely with WIPI2, and you already see that WIPI3, and in particular WIPI4, looks a bit different. And we went on to address whether or not they're redundant or different in their function in autophagy, and first downregulated them in cell lines that express similar levels of all 4 of them. And what you can see here is the control in autophagosome, double-membraned vesicle. And in the absence of WIPI4, we see reduced numbers, but they can still form. In the absence of WIPI2, which is an essential ATG protein, we saw the appearance of premature structures. But with WIPI3 and WIPI4, which is of our interest today, we saw elongated early structures. So this picture already indicated that they may not be redundant in their function autophagy. But this really then became more clear when we did conducted 2 unbiased approaches. One, where we addressed the WIPI protein interactome, and what you see here is they all have different partners. In fact, we could only verify one partner that is overlappingly bound to by WIPI1, 2, and 4. And all others are individual partners. And in addition, we conducted a kinome screen where we addressed the regulation of WIPI function. And to be not too long in my discussion of this, I present you the summary of this study in this scheme, which is our current model. So you see here a WIPI circuit, and we believe they work really in a circuit, all as PI3P effectors at the nascent autophagosome. But on top of this, we have WIPI4 being involved in a signal cascade, and we saw that WIPI4 binds to AMPK and also binds to its intimate partner, which is ATG2. And we have evidence to believe that upon AMPK activation, when energy is low in the cell, the WIPI4 ATG2 complex translocates to the site of autophagosome formation and controls the size of the growing phagophore. Now, as we've heard before, mutation in WIPI4 or WDR45 were identified as being causative for BPAN, and this, and you're all aware of this, is due to the fantastic work published in 2012 by Susan Hayflick. And later on, this was confirmed by many other groups. But recently, it was also shown that mutations in WIPI3 and also in WIPI2 lead to problems with intellectual capacity. So we feel that this whole family is very interesting with regard to neurodegeneration. So what I'd like to compare here is the first assessment of BPAN patients by Saitsu et al., and this is the publication from 2013, where they showed that autophagy is blocked using LC3 lipidation assay and also looking for LC3 puncta per cell. So this indicated that the mutation causes a block in autophagy, so autophagy is altered. And this compared nicely to the result we had on WIPI4 knockdown in human cells, where I showed you already that early structures elongate. This is also shown here in high throughput analysis. And when we knock down WIPI4 in human cells, we again see that autophagy is blocked by, this is shown here by the accumulation of GFP LC3 puncta per cell, and also in this lipidation assay. So what is shown here is again the WIPI4 beta propeller. We defined one amino acid that is crucial for AMPK binding. We defined two essential amino acids required for binding to ATG2. And here you see on the other side of the propeller, which is here, it's a docking experiment with PI3P. So based on these results, we felt fairly confident to start a new project trying to identify what is wrong, or how does autophagy function differently in BPAN patient. And this was really initiated by Matis Zinovcik, who found in one of his patients a novel mutation, and that was a mutation in an intron. And what you see here is the result of this mutation is basically the deletion of exon 4 and the insertion of an intronic sequence. And this that this occurs is verified by sequencing, also by PCR. What you see here is in the fibroblasts that were then isolated from patients and also from the mother and the father, that the wild type sequence, this is mRNA abundance, drops by 50% as expected. We then went on and did very basic autophagy assessments. And this is an LC3 lipidation analysis where we could confirm these are three controls that LC3-1 and LC3-2 accumulates, indicative, like I've shown before, for a block in autophagy. However, we felt it would be necessary to go further and address this also in an imaging approach. And this is an automated analysis here, where you see in fed and starved conditions, isolated fibroblasts from the subject, the mother and the father, and then an age and gender matched control. And we stained for endogenous P62. It's a selective, it's a marker for selective autophagy, the first autophagy receptor, in fact, that was identified, and is used to address the autophagic flux. And additionally, we stained for endogenous WIPI2. You also see the nuclei and the cell outline. And in the quantification here, you can appreciate that P62 is accumulating in the subject. And this is indicative for a block in the autophagic flux. So not only the accumulation of early membranes, but really a block in the flux. Likewise, the numbers of WIPI2 puncta per cell decreased significantly, and those puncta show autophagosomal membranes. So we have less autophagosomal membranes and less autophagosomal autophagic flux. And we also addressed with this automated system the rare event of P62 and WIPI2 co-localization, and that also dropped. So we then were interested to see what kind of putative protein could be still present. Let me just flip back, and I will go further quickly. So this in this patient, we would have a premature stop codon, but still a peptide could be expressed, and it is assumed that this is degraded, but it may not be degraded. So we were interested by homology modeling how this may look like. So this is the wild type propeller of WIPI4 and depicted in pink, the PI3P binding site. In blue, this is the unit that is still present in the mutant. And then in after this, this darker brownish part, it would stop. And when we just look at this part, you see that in the patient, no propeller would form, at least in those cells that express the mutant. So there's no way that this fragment could bind PI3P, and it may be degraded. We cannot detect it on normal protein gels, but it may, and this is a speculation right now, it may fulfill a new function. And this is our interest in going further with this. This is not our scheme, this is from this review here, as the speakers that will follow, Robin and also Mario, we will go ahead to address the question, what is the mutated WIPI4 WDR45 function in patient-derived cells? And we will be using and have already established fibroblasts and iPS cell lines. We'll differentiate them, and we aim to address the question, what is different with regard to autophagy, and which other non-autophagy function the new forms may fulfill. And this brings me to the most important slide, and this is my lovely group. And I showed you results from David Schüssele today, Katharina Sporbeck, and Maximilian Haas. And I would like to stress our wonderful collaboration with Matis Synovczyk from the Hertie Institute. And I'm extremely excited about our cooperation with Sarah Griesholm from the States, from BPAN Worries. We had fantastic conversations in the last months, and with Marcus and his family as well. And I'm very happy to take questions if I'm not too late. I stop sharing my screen now.
We are behind the schedule, so maybe I will read only one question. It's from Susan. Can you infer a functional order of the YPI proteins? For example, YPI2 function seems to depend on what YPI4. Can you order or sequence the orders?
Yes, so we and others have evidence that YPI2 is the factor that is required for LC3 lipidation. We have evidence that YPI1 assists in this function, and then downstream LC3 lipidation occurs, and downstream of this there's a role for YPI4 and also YPI3 in regulating the size of the phagophore. And recent data by a couple of groups have nice evidence and suggest that the complex of YPI4 and ATG2 is there to transfer lipids, for example, from the endoplasmic reticulum to the growing phagophore. And that's, in particular, it's interesting because ATG2 also localizes to lipid droplets in addition, where we, and this is unpublished as well, also find YPI4.